This video is part 1 for subtopic 2.5, enzymes. Here's a list of understandings that will be covered in this video. The other understanding, application, and skills will be covered in a separate video. So we'll start with just an overview of enzymes. So all chemical reactions do require an initial energy input to get started, no matter what they are. So in thinking about a reaction in which reactants turn into products, reactants need to have bonds that become broken. Molecules need to reorient themselves, and then at the end of it, new bonds need to be formed. So in order to create a new molecule, new bonds have to be created. So this initial input of energy into the reaction is what's referred to as the activation energy. This is the energy needed to get the ball rolling for that reaction to occur. Where enzymes come into play and where enzymes are used is that they are used to catalyze or speed up chemical reactions in cells. So enzymes make reactions go faster within cells. So catalysts will speed up the rate of a chemical reaction without themselves being used up. So an enzyme will be will react with a reactant, or will fit with a reactant to convert it into a product. And at the end of the reaction, the enzyme will be available to perform that reaction again. So catalysts speed up spontaneous reactions by reducing the activation energy. So that initial input of energy to make a reaction happen, enzymes work by reducing that energy that is needed to make a reaction happen. So this can be illustrated in the following energy diagram, in which we have the progress of the reaction. So this is as any reaction is occurring. And then we have the energy content of the molecules. So there's two lines on this diagram. There's a black line and a red line. The black line, which I'm outlining in blue, is the course of a reaction without a catalyst. And this peak here represents the activation energy required for this reaction to happen. So there has to be an input of energy to reach a certain threshold, and then once that threshold is reached, then the reaction can happen. In this case, the reactants that we begin with versus the products that we end with, the products have a lower energy level than the reactants. So there's a decrease in energy content as we go from reactant to product. The important thing that catalysts do is this activation energy that can be quite high for some reactions. Enzymes will actually decrease that activation energy and help speed up the rate of that reaction. So the red line, which I'm outlining in green, would be the activation energy that is needed with a catalyst, or that activation energy that is lowered with the presence of a catalyst, such as an enzyme. So enzymes work by making reactions happen faster by lowering the amount of energy that's needed to get them going. That's how a catalyst works, and an enzyme ultimately works. So we'll get into some of the specifics in which enzymes work and how they work to speed up reactions that occur. So enzymes have what's called an active site, which will bind to a specific substrate. So in the diagram that's shown here, so it shows both enzyme, active site, and substrate. So the arrow is the substrate, and the substrate we can classify as the reactant in a biochemical reaction. The enzyme is a globular protein which acts as a catalyst for biochemical reactions. So that enzyme is going to take that substrate and turn it into a different product or a different molecule. So it'll do that by breaking big molecules into smaller molecules, or taking smaller molecules and making them into bigger molecules. Now, every enzyme will have what's called an active site. In here, it's the active site I've outlined in green. And that active site is the region on the surface of an enzyme to which substrates bind and which will then catalyze the reaction. So the active site is where the substrate will attach to the enzyme, and then at that active site, this is where the reaction will occur. So the steps that are involved in an enzyme catalyzed reaction is that the substrate will have to come into contact with the active site of the enzyme. The substrate will then bind to the active site on the enzyme. This will create an enzyme substrate complex. And then a reaction will occur, and in this case, we have a large molecule changing or reacting to become a smaller molecule. So a reaction will occur through that active site, which will now change our molecule. And here we see it's caused a reaction in which it breaks apart that large molecule into smaller molecules. The products then get released from the active site to be used elsewhere within the cell. Now, the enzyme is unchanged at the end of this reaction and can be utilized over in this process. So it's ready to bind more substrate and perform this reaction again. So one of the important things to keep in mind with the active site on an enzyme is that the shape of the active site is complementary to the shape of the substrate. So in this case, the shape of the substrate is an arrow shape to it, and so the active site on the enzyme will have a shape that is complementary to that substrate, which is what allows it to bind. So there are a few different models for describing this enzyme specificity that occurs. So the first model is referred to as the lock and key model. So you basically have a key that will fit a particular lock, and only that particular key can fit into that lock to create a lock-key complex. So much the same way an active site has a specific shape to it, that will only fit a specific substrate, which will create an enzyme substrate complex. So the substrate and the active site must match each other in two ways. So there must be a match structurally, where the three-dimensional structure of the active site is specific to the substrate. Substrates that don't fit won't react. The substrate and the active site have to match each other chemically. So substrates that are not chemically attracted to the active site won't be able to react. So if there are similar charges between the substrate and the active site that would cause repulsion, you won't get a reaction that occurs. And the diagram at the bottom just shows this lock and key model in action. So here we have the enzyme, and we have the substrates. So this is the E, this is the S. The substrate fits into the active site, or is specific for that active site, forming an enzyme substrate complex. The reaction occurs where that substrate is turned into a new product, and we now have an enzyme product complex. So the product that is just being created is still within the active site. That product gets released from the active site so that we now have a free enzyme and we have free product. This enzyme is now available to start this reaction over again and bind another substrate. The lock and key model is a good model to explain enzyme specificity, but it doesn't take into account how enzymes might be able to bind multiple substrates. So here we have the induced fit model for enzyme specificity. And the in the induced fit model better explains enzyme activity. So it turns out that if the lock and key model were true, one enzyme would only catalyze one reaction. In actuality, enzymes can catalyze multiple reactions. So there can be multiple substrates for one enzyme. So the induced fit model better explains how one enzyme is able to catalyze reactions for multiple substrates. So the way the induced model works is that if we have our enzyme, here we have our active site on the enzyme, as the substrate, and in this case sucrose, comes into contact with the active site and binds to the active site now changes its shape to better fit the substrate. So in a way, there is a conformational change in the active site to better fit that substrate. So you can almost think of the enzyme hugging the substrate as it comes into contact with it. It hugs it so hard that it causes a reaction to happen. And in this case, we have sucrose that gets hydrolyzed or split into its monosaccharides. So that sucrose gets split into fructose and glucose, which then gets released from the active site of the enzyme. The active site changes back to its original shape and then is ready to be used again. So with enzyme catalysis, this doesn't involve molecular motion and collisions of the substrate with the active site. So in order for enzymes to function, we have to keep in mind that an enzyme is in solution or in that cytoplasm, kind of moving around. We have the substrate in the cytoplasm moving around, and enzyme and substrate have to come into contact with each other. And so the coming together of a substrate and an active site is known as a collision. Like these things like literally collide with each other. Most enzyme reactions, as I mentioned, occur when the substrates are dissolved in water, and all molecules that are dissolved in water are in random motion. So things are just kind of zipping about. So collisions are the result of that random movement of both substrate and enzyme. So when a collision results in a reaction, it is considered a successful collision. So a successful collision is one in which substrate and active site correctly align to allow binding to take place. If the active site is has a specific shape for a particular substrate, then the substrate has to align properly with the active site in order for binding to occur. So this is illustrated on this particular diagram, in which we have the substrate maltose and the enzyme maltase. So here we have a very particular shape for this active site on this enzyme. And for a reaction to happen, the substrate, in this case maltose, has to come into contact in the correct orientation with the active site in order for a reaction to happen. If it comes in at an incorrect orientation or an incorrect direction, then no reaction would happen. So there are a number of conditions in which enzyme activity can be affected, which include temperature, pH, and substrate concentration. So the first that we'll look at is the effect of temperature on enzyme activity. So here we have a graph that demonstrates what happens to enzyme activity as temperature increases. So on our x-axis, we have increasing temperature. On our y-axis, we have the rate of enzyme activity. And so obviously the higher up you go on the y-axis, the more activity you have from your enzyme. So there are several points to note that indicate what is happening to the enzyme activity. At point A, where the temperature is rather low, and as the temperature is increasing, and when it reaches point A, that increase in temperature increases the kinetic energy of the substrate and enzyme. With that increased kinetic energy, you increase the random motion of both enzyme and substrate. And so you get an increased chance of collision and reaction. So this causes an increase in the rate. So low temperatures tend to have a low rate of reaction because substrate and enzyme have a low amount of kinetic energy. So as the temperature increases, the rate of the reaction increases. As these molecules have more energy, they move faster and therefore collide and react more frequently. So as the temperature continues to increase, we reach a peak point. And this peak point, which is point B on the graph, is referred to as the optimum temperature. And the optimum temperature is where we get the maximum rate of reaction. So this is the maximum amount of activity that we have occurring for our enzyme. And here we have a balance between the enzyme stability and the kinetic energy of the reactants. We want to continue giving our reactants more energy, but then we have to keep in mind that, as the temperature increases, we can potentially, or we are starting to disrupt the R group interactions of the enzyme. And so as you keep increasing temperature and you go beyond the optimum temperature, so above that optimum temperature, further increases in temperature will lead to the denaturation of the enzyme. As the enzyme starts to denature and starts to unfold because of disruption in those R group interactions, the active site starts to change, and so we'll lose function. And as temperature continues to increase, you'll see that there is a rapid drop in enzymatic activity. So at these high temperatures, this destabilizes the enzyme molecule, and basically at this point here, the enzyme is denatured, which is why you see this very rapid drop in enzymatic activity. Now there is a type of bacteria referred to as a thermophile, which is found in deep sea vents. And this particular bacteria is able to withstand much higher temperatures before its enzymes will denature. So, temperature can be a very enzymatic specific thing. Not all enzymes denature at the same temperature. So one of the important things that you should be able to do is you should be able to sketch this graph and point out some of the important parts of what's going on here. So, where the optimum temperature is, why an increase in temperature beyond the optimum temperature causes such a rapid decrease in enzymatic activity. So the next effect we'll look at is the effect of pH on enzymatic activity. And when we look at pH activity, so as we increase pH, so if we're increasing pH at the lower end, this at the lower pHs we'll have more acidic conditions, and as we increase pH, this will create more basic conditions. We can also compare that to the rate of enzymatic activity on our y-axis. So you'll notice the shape of the curve for pH is that there is a very steep increase till you reach like an optimum pH, and then a very steep decrease. So ultimately, what this curve is kind of suggesting is that enzymes operate within a very narrow range of pH values. So if we look at what's going on in our graph, so we can start at point A. Basically, when you have a decrease in pH, you are increasing your hydrogen ion concentration. These hydrogen ions can interact with the exposed R groups on the active site, and this will ultimately cause the active site to change shape as those R group interactions are interrupted. And this will reduce the specificity of the enzyme, or the ability of the enzyme to bind to substrate. So you get a decrease in the rate of reaction. So as conditions become more acidic, that rate of reaction drops off dramatically. At point B, this is where we have the optimum rate of reaction for this particular reaction occurring. And this relates to point D on the graph as well. So at this point, the active site structure and structures specific to the complementary shape of the substrate are intact. So you can get successful enzyme substrate complexes being created, and therefore reactions can occur. And then as you move beyond the optimum pH, which is point C, this is where we get an increase in pH. And so as we increase the pH, we get a decrease in hydrogen ion concentration or an increase in base concentration. So we're increasing hydroxide ion concentration. This has the same effect as decreasing pH. Ultimately, that active site changes shape. We get reduced specificity and ultimately a decrease in the rate of reaction. So for both of these points, as we deviate from the optimum pH, basically the hydrogen bonds between those amino acids in the structure of the enzyme become broken. This results in a loss of shape of the active site of the enzyme, and so it does not function. As a result, this is generally a permanent change. So once you go beyond the optimum pH, you get that denaturation occurring, and then if you try to return to the optimal pH, you do not regain enzyme activity. That enzyme activity is lost. So in this, on this slide, these two pictures are showing that optimal conditions can be different for different enzymes. So one figure is showing a change in temperature and a rate of reaction, and it's showing that for two different enzymes, there can be different optimal temperatures that can exist. So for example, here we have the optimal temperature for a typical human enzyme, which is somewhere around 37 degrees Celsius, whereas the optimal temperature for an enzyme from these thermophilic bacteria, these bacteria found in these deep sea vents, they have to be able to tolerate these high temperatures in order to live next to these deep sea vents. So their enzymes denature at higher temperatures. And here we see that the optimal temperature is around like 75, 76 degrees Celsius. So those optimal conditions can vary depending on the enzyme. pH can also, optimal pH can also vary depending on the enzyme as well. So enzymes found in the stomach have to be able to tolerate those acidic conditions, so they have an optimal pH probably closer to 2, which is typically the pH of stomach acid. Whereas enzymes found in the intestine have to have a higher optimal pH because those conditions tend to be more basic. So the last effect that we'll look at is the effect of substrate concentration on enzyme activity. And here we have a figure demonstrating what happens as substrate concentration increases versus the rate of enzyme activity. So at point A, as concentration of substrate molecules increases, the chance of collision with of substrate with the enzyme increases as well. So we're creating a greater chance of forming this enzyme substrate complex. And so as a result, we see an increase in the rate of reaction. So increasing substrate concentration generally will increase the rate of reaction. And so as we continue to increase substrate concentration, we reach this point B where we start to see the rate begins to level or begins to plateau. And at this point where our active sites on the enzymes are becoming saturated with substrate, so all of the available active sites from all of the enzymes in solution are becoming fully occupied. So basically, new substrate must wait for a previous reaction to complete and the product to exit the active site. And once that active site is now open, then another substrate can bind. So basically, you can kind of think of it as all the enzymes are busy. Any substrate in solution has to wait until an enzyme becomes available. And then beyond this point B, as we continue to increase substrate concentration, we see that there's really no significant increase in the rate of enzyme reaction, because at this point now we have full saturation of the active sites by the substrate. So at this point the rate becomes constant. So the rate of our enzyme activity does not change. And the rate is constant for any further increases in substrate concentration. So any increases in concentration beyond this optimum value will have no added effect, as there are no extra active sites to be used. So as with the previous graph with pH, you should be able to sketch this graph and describe what is happening as changes in substrate concentration are made or changes in pH are made. So in this last slide, just very quickly, this relates to the denaturation of proteins that was discussed in 2.4. But enzymes, because they are proteins, can be denatured as well. So with that denaturation, we have a loss of R group interactions. So if this is our active protein, which is functional, it can lose its structure. So in that functional protein, it has a tertiary structure to it, and in a denatured protein, there is that loss of the tertiary structure as well as the secondary structure. All that remains is the primary structure, where we just have amino acids linked together in that chain. So the remaining diagrams illustrate what happens with an enzyme as denaturation occurs. So if we have our enzyme with its active site before denaturation, as it loses, as those R group interactions become disrupted, there now becomes a change in the shape of the active site, and this active site can no longer bind substrate. So in the following diagram here, we can see the active site that's able to nicely bind its substrate, while the denatured enzyme really loses its shape, and that substrate can't fit anymore. And when that substrate can't bind to the active site anymore, there is a loss of enzyme activity, or reactions cannot occur anymore. So this is the end of part 1 for subtopic 2.5 on enzymes.